A very good morning to all. Today's topic is herpes viruses. Herpes viruses are included in the family Herpes viridae, which has been divided into three subfamilies. Let's see classification. Classification of human herpes viruses. In these human herpes viruses, we can see three types of the families: alpha herpes viridae, beta herpes viridae, gamma herpes viridae. In alpha herpes viridae, we can see human herpes virus 1, human herpes virus 2, human herpes virus 3. In the beta herpes viridae, we can see human herpes virus 5, human herpes virus 6A, human herpes virus 6B, human herpes virus 7. In gamma herpes viridae family, we can see human herpes virus 4, human herpes virus 8. In common names, we can see herpes simplex virus 1, virus 2, varicella zoster, herpes B virus or herpes virus simile. Beta herpes viridae, we can see cytomegalovirus. Gamma herpes viridae, we can see Epstein-Barr virus. Third one is morphology. This is herpes virus. Size 100 to 200 nanometer. Shape roughly spherical. Symmetry icosahedral. Envelope present. Nucleic acid double stranded DNA. These viruses contain DNA genome. The core is protected by capsid and it is composed of 162 capsomers. An outer layer envelope containing peplomers, 162 peplomers. Between the capsid and envelope, tegument, a protein structure. This is tegument, a protein structure is present. Next one is cultivation. The herpes virus can be cultivated on chorioallantoic membrane, rabbit kidney, HeLa, human allantoic on tissue culture. After incubation, the virus produces cytopathic effect and some viruses show inclusion bodies. The virus multiply in nucleus of the host cell producing Cowdery type A intranuclear inclusion bodies. Next one is resistance. The herpes virus can be destroyed by heating and remaining viable for yeast at low temperature. The viruses are sensitive to alcohol, ether and chloroform. We should know about alpha herpes viridae. Viruses of this group have a wide host range of humans, cause latent infection in sensory ganglia. They have a short replication code of 12 to 18 hours. This subfamily contains three important human pathogens: HSV1, HSV2, varicella zoster. Herpes simplex virus. It is a very common human pathogen. There are two types of herpes simplex viruses, HSV1 and HSV2. HSV1, it causes infections of mouth, eyes, CNS, etc. Example, gingiva stomatitis. Ulceration of mouth, tongue, gums, and the patient experiences pain and bleeding of the gums. It is commonly seen in primary herpetic infection. Herpes labialis. Sore of cheek, chin, upper lip, nasal fold or inside the nose. Eczema herpeticum. It is usually limited to skin but may spread to liver, spleen, lungs and rarely to CNS. Keratoconjunctivitis, inflammation of cornea. In HSV2, it causes general herpes. Genital lesions occurs on the urethra, cervix, penis and vagina. Neonatal herpes. Infection seen in premature infants. The infection will spread throughout the body to many organs. Let's see differences between HSV1 and HSV2. In HSV1, it is cultivated in chick embryo chorioallantic membrane. HSV1 produces smaller pox. On chick embryo cam, HSV2 produces longer pox. Sensitive to two antiviral agents, cytorabin. Resistance to antiviral agents, cytorabin. Exhibit less neurovirulence in lab animals, exhibit more neurovirulence laboratory animals. Let's see pathogenesis. Virus route of entry is skin, mucous membrane to mouth or eyes. It multiplies and it passes into the lymph nodes. It causes lymphadenitis. And HSV travels to the nerve root near the spinal cord and settled here. And it passes, integrates into the host cell genome. From these, HSV in the latent state may get reactivated by the various stimuli like cold, fever, stress, exposed to sunlight. And its recurrence is more frequent. Next one is pathogenesis. In pathogenesis, we can take samples, vesical fluid, skin, swab, saliva, CSF, corneal scrapings, etc. In direct microscopy, smear preparation from the bone vesicles and stained with 1% aqueous solution of the toluidine blue. Examine under microscope. Multinucleated giant cells observed. In this, we can observe cytopathic effects which produces in the cultivation of herpes viruses. Next one is inclusion bodies. Smear preparation stained with Giemsa stain. Observe under microscope. We can see in this process inclusion bodies. As Cowdery type A, intranuclear inclusion bodies are observed. In electron microscopy, smear or examined under electron microscopy for the presence of virus particles. In immunofluorescence staining, it is used to observe the presence of viral antigens in scraping and tissue preparation. In serology tests, we can take for the detection of herpes virus. We can take ELISA, enzyme linked immunosorbent assay, CFT, neutralization test, RIA, radioimmunoassay are employed for the antibody detection. PCR, polymerase chain reaction can be used for the detection of HSV DNA. In chemotherapy for eye and skin infections caused by the HSV1, acyclovir in the form of the ointment can be used. For HSV type 2 infections and famciclovir are more effective oral agents.